Okay, so these are the things that you're going to want to know. Some examples of prokaryotic and eukaryotic organisms. You maybe can already check that one off your list of things to do. Compare and contrast prokaryotic and eukaryotic cells. How are the size differences among these things, and why are cells usually little? Okay, and then if you guys are keeping a little notebook or whatever of the root words that we talk about, you're going to want to add eu, pro, and carry to those if you don't already have them in there, because this will help you put context onto unfamiliar words. Okay. So remember, like the first or second day of class, we played this game that my kid made up. I call it "There's a Hammer in My Beer." So we're just like kind of going back to that. So, you know, we can have complex organisms that are made of organ systems, that are made of organs, that are made of tissues, that are made of cells in our multicellular organism. Or we might have a single cell organism, which the organism is the cell, right? So those things are the same if you're just one single cell. And that's true for all prokaryotic organisms, and some eukaryotic organisms just chill all by themselves as one cell. Okay, so just kind of some historical context behind our unified cell theory. You thinking back to what a scientific theory is. I Googled how to say this guy's name and I forgot. Yep. Anyway, so this Dutch dude made the first microscope, and as he was like, oh my God, there's all this stuff that's really little that we didn't know about before because it was too little to see with our naked eye. And so when we use our microscopes, we have a little power switch so we can turn the light on. But in these old timey ones, there's just a mirror. So you kind of have to like make sure that you got a nice strong light source, and you got to angle that mirror so that it's catching the light and reflecting it up into the series of lenses and stuff that magnifies the thing that you're looking at. So we first got this term cell in a paper that was published way back in 1665 by Robert Hooke. He was looking at cork, which is plant cells, under this microscope, and he's like, oh, look at all those little rings. It looks like jail cells. So boom, now that's why we call them cells. And then technology improved. We're able to refine how those microscopes are used. And then we were able to see even smaller things, specifically single cellular things in prokaryotes. So bacteria is a prokaryote, protozoa, or eukaryotes. Single cells, they were discovered then. And then these other three dudes, they were kind of working on these defining a unified cell theory independently, and then they were like, hey, we should collaborate because we're doing the same stuff. I was like, yeah, that's a good idea. And so they came up with a unified cell theory. And remember, somebody remind me what a scientific theory is versus like, you know, like, oh, I've got a theory about the upcoming election. That's a hypothesis. A series of very well tested hypotheses that stand up to time, right? So people tend, like normal use of the word theory, people tend to mean a hypothesis, but when we're talking in scientific terms, a hypothesis is kind of the first step. It's kind of the first stab at answering a question. A theory, on the other hand, it's been tested over and over and over and over and over and withstood that test. Okay, so here's our unified cell theory. There's three parts. Part one goes like, in order to be a living thing, you got to be made of cells, at least one cell. If you don't have a cell, then you're not alive. Haha, viruses, you're not alive because they don't have true cells. So there you go. That kind of leads into the second point. Cells are the smallest unit of life. You can take a multicellular organism and break it down, break it down, break it down, break it down. But until you get to the cellular level. At the subcellular level, you start to get into this non-life thing, right? Because you have to have all the parts of the cell. And then kind of Louis Pasteur is the dude that kind of came up or like contributed this part. New cells are formed only from pre-existing cells. We don't have spontaneous generation. Life doesn't just come about, right? We have to have other cells that reproduce to make new cells. And so I've got a picture of some plant cells. These are some red blood cells, and these are some bacteria in our little pictures there. Okay. All right, so we already know that our two biggest classifications are prokaryotic cells, eukaryotic cells. Pro means what? Before. U means true. Carry means. So we can like kind of put those words together. These are before, these cells were before we had a nucleus. These are cells with a true nucleus. Okay, this is kind of review at this point. All right, so in Venn diagram world, because there's lots of Venn diagrams that's gonna happen in this chapter, you guys. In Venn diagram world, these are the things that go in the overlap. All cells have a plasma membrane. We spent a week talking about how important plasma membranes are. All cells have cytoplasm. This is the cell juice. This is the goo that all the DNA and the organelles and all that stuff, the proteins and the solutes and all that blah, blah, blah stuff that we've been talking about is floating around in. We gotta have DNA. If you don't have DNA, you're not alive. Must have DNA. And then you also have to have this organelle called ribosomes. Ribosomes, does anybody know what ribosomes do in the cell? That's the site of protein synthesis. So if you can't build proteins, you're not alive. Okay, so here's our plasma membrane in our eukaryotic cell. It's this little double line here. In our prokaryotic cell, it's this little line here, and then we've got this extra stuff on the outside of that. We'll talk about that later. Cytoplasm, that's just the goo that everything else is suspended in. DNA, eukaryotic organisms, it kind of hangs out here in the nucleus. In our prokaryotic cells, it's just one circular chromosome. And then ribosomes, that's indicated by these little dots. Sometimes they're stuck to this thing called the endoplasmic reticulum. Sometimes they're just chilling all on their own. And even though prokaryotes don't have a lot of organelles, they all have ribosomes. Those are those little dots. Cool. All right. So in order to understand why cells tend to be small, we have to understand this concept of surface area to volume ratio. So in order to really understand what's going on, we're going to do some math. It's going to be fun. So we're going to stop here for a second. We're going to go back to the lab. I have, there's some handouts over there. Basically what we're going to do is we're going to measure a big cube and a little cube, and we're going to compare their surface area to volume ratio. And then we're going to take our little cubes and we're going to understand, we're going to explore shape. So we're going to compare our little cube surface area to volume ratio to a marker. So basically, let's see, I can't remember what the next thing is. Okay, yeah. So let's stop there. We'll go migrate back to the thing, to the lab area, and we'll work on this for a second.